In this video, we are working toward drawing abundance versus pH diagrams for amino acids and even proteins. We will start here by identifying fully protonated forms of amino acids, writing acid dissociation reactions for amino acids, and learning how to identify the isoelectric form of an amino acid. So as a reminder, you know the neutral amine carboxylic acid form for amino acids with the alpha carbon in the middle. And you've also become familiar with the charged or beta-ene form where the amine has turned into an ammonium and the carboxylic acid has turned into a carboxylate anion. Now we're going to introduce the fully protonated form of the backbone of an amino acid. Here you see that the amine is an ammonium, so it's the conjugate acid form of the amine group, and the carboxylic acid is in its most acidic form, so it's the conjugate acid rather than the conjugate base form that would be given by this carboxylate ion over here. That's the fully protonated form because every acid or base group is in the acidic form, is in the weak acid or conjugate acid form. There's also a fully deprotonated form of the backbone where every acid or base group is in its fully deprotonated form. So the amine is present again. It's lost its extra H plus that it had in the conjugate acid ammonium ion, and the carboxylate ion is present. It's lost the H plus that it had as the carboxylic acid group. It's important that you become familiar, that these four start to look really different to you, and that you can notice which one you have in a given situation. I'll also call your attention to the fact that in this case, the fully protonated form has a charge of +1, and the fully deprotonated form has a charge of -1. That won't always be true, depending on what the side chain does, but it is important to see that the charge changes as you change the protonation state of the various acidic and basic groups in the amino acid. So the purpose for looking at that fully protonated form is that weak acid reactions of amino acids always start from the fully protonated form. So in the most acidic environment possible, this is the form that will be present for the amino acid. And for the sequences of reactions that we'll draw, this is where you always need to start, with all the groups fully protonated in their conjugate acid forms. So that's the fully protonated form. Remember, it had a plus charge. And as all acids do, it reacts with water, and acids donate H plus to water. So the question is, which acid group will donate its H plus? And you can see that what happens is the carboxylic acid donates its H plus first and makes the H3O plus. The conjugate acid-conjugate base pair is shown here. Of course, there's also a conjugate base, conjugate acid pair between water and H3O plus as well. And the way we know which group is going to lose its H plus first is that we can look up the pKa for the various groups. And the pKa's for backbone carboxylic acids, also denoted alpha CO2H, range from 1.7 to 2.6 across the 20 amino acids that we're looking at in this course. That pKa controls the crossing point for this conjugate acid-conjugate base pair on an abundance diagram. So that's a little hint about drawing abundance diagrams that you're going to need later. This is a lot to write out, especially when you're drawing large structures with the full side chain. So we've developed a schematic representation shown down here at the bottom. You can see that you still have the fully protonated form and the form that has lost one H plus from the carboxylic acid to make the carboxylate ion. But instead of showing the reaction with water, we simply show the loss of an H plus going this direction and the gain of an H plus to go from right to left on this reaction equilibrium arrow. The charge does change because you lose an H plus, you lose one positive charge on the molecule and go from a +1 to a neutral species. From that neutral species, the beta-ene form that we had in the past slide as our conjugate base, we can actually lose another H+. So this is still a conjugate acid form of a weak acid, and it can donate its proton to water. Again, it's in equilibrium, and we get the amine and H3O+. So now we're in the fully deprotonated form of the backbone. And you can see that by losing a positive charge as an H+ from this neutral species, we end up with an actual negative charge species. The formal charges add up to that -1. The pKa's for backbone ammonium ions, also known as alpha NH3+, range from 8.8 to 10.8. So they're quite high, and that provides the crossing point for the conjugate acid conjugate base pair that we have above, from the beta-ene form to the fully deprotonated form of the backbone. The schematic representation for this, very similarly, you take the full form for the amino acid, but you just show it losing an H+ to make the next conjugate base down. So the ammonium ion, has lost an H+ to form the amine. The carboxylic acid had lost its H+ in the previous step, so it's just carried along for the ride in this picture. And if we wanted to go back the other direction, we'd be adding an H+. So test yourself, please. Write the fully protonated and fully deprotonated forms for alanine. Pause the video and actually write those out. Okay, hopefully you were successful with this. Here is the fully protonated form, carboxylic acid, and the ammonium ion. And because there's 1+ charge, formal charge, the overall charge on this amino acid form is +1. And the fully deprotonated form, we've lost the H+ from the ammonium to make the amine, and we've lost the H+ from this carboxylic acid to form the carboxylate ion. And the net charge is -1. So now I would like you to test yourself again, starting with the fully protonated form that you just drew for alanine. Write the full reactions, so react this with water, and write the schematic reactions for alanine. And I'd like you to try to label the isoelectric species. The isoelectric species is just a fancy word for the species that doesn't have a charge. You will need a couple of pieces of information. The alpha ammonium ion, the pKa for that proton is 9.69, and the alpha carboxylic acid's pKa is 2.34. Again, the alpha refers to the backbone ammonium and the backbone carboxylic acid, because in some cases side chains will have carboxylic acid or ammonium groups, and so we'll need to be able to differentiate those. Go ahead and pause the video and try to write that, write those reactions. All right, here's the first reaction from the fully protonated state. Alanine is going to act as a diprotic weak acid, sort of like H2CO3 would. It's going to lose one proton, and then it'll lose another proton from the conjugate base of that first reaction. So here's our first reaction with water. The pKa is 2.34, and you can see that that is the carboxylic acid H+. It's lost to form the carboxylate ion and H3O+. Elements you should pay attention to: did you put brackets around all charged species with the charge, the net charge outside without a circle, and any formal charges inside with a circle? Did you use an equilibrium arrow? Did you write the pKa above the equilibrium arrow? If you did, you're doing a good job. Next, we have the reaction of this first conjugate base as another acid. So that's the same species down here, and the carboxylate has already been formed from the carboxylic acid. Now we still have this ammonium group with a pKa of 9.69, so that will react with water to give an H3O+ and the amine left. The schematic that you would need to draw for our exams is starting with the fully protonated form, lose the first hydrogen with a pKa of 2.34, that's a hydrogen ion, an H+, and form the carboxylate ion, and the ammonium ion is carried along from the previous form. The charge went from a plus to a neutral because this H+, which left from the carboxylic acid, forms the carboxylate ion with a negative charge. Now this species will actually lose another H+ from the ammonium ion to form the amine, and the net charge at this point is -1. There's no charge left on the ammonium, and we still have the charge on the carboxylate ion. The pKa for that is 9.69. A rule of thumb is that in weak acid reactions, the H+ that has the lowest pKa always reacts first, and in fact they'll react in order from lowest pKa up to highest pKa. So just a review of that schematic for the exam. The fully protonated form, we lose the lowest pKa H+ first, that's the carboxylic acid one, and then in the next reaction we lose the pKa of 9.69 ammonium ion and turn that into an amine. We're going to label these A, B, and C just to be able to refer to them more easily. I don't have to keep saying the fully protonated form. So the fully protonated form is A, the beta-ene form here is B, and the fully deprotonated form is C. In this example, the one that doesn't have a net charge is B, and that makes it the isoelectric species. That's how you find the isoelectric species. It's the species that doesn't have a charge. It won't always be B or the second species. There are 13 amino acids that have side chains that aren't acidic or basic. So they behave just like alanine, which we just looked at. No big surprise here, leucine is one of them. There's no groups on this that could do anything. The threonine has an alcohol group. That alcohol group acts as a molecule. It doesn't react as an acid or a base. And there are 13 total side chains that act this way. There was a little test yourself there. Can you draw the fully protonated form? I drew the beta-ene form, which you're used to from memorizing them. But the fully protonated form has that ammonium group in the conjugate acid form and the carboxylic acid in the conjugate acid form, and as a result it has a net charge of +1, and we had to put the brackets on. So go ahead and pause the video and try to do that for threonine. I hope that's getting pretty easy now. Carboxylic acid and the ammonium, so the fully protonated forms of both acidic and basic groups in the side chain. 7 amino acids have side chains that have weak acids or bases in them, or that act as weak acids or bases. No big surprise, aspartic acid is one of them, and histidine is one of them. So go ahead and try to draw the fully protonated forms of both of those species. Here is aspartic acid. The fully protonated form has the carboxylic acid in the backbone, the ammonium acid in the backbone, and the fully protonated carboxylic acid rather than the carboxylate ion in the side chain. The result is this is neutral, and this positive formal charge makes the whole species a +1. So the fully protonated form is positive for aspartic acid. For histidine, we have the carboxylic acid as always, the ammonium ion as always, so that's what our backbone always looks like when it's fully protonated. But we also have an extra H+ in the histidine ring, and it attaches itself to the nitrogen that used to be an imine with a lone pair, and we get a positive formal charge and 4 bonds to that nitrogen. The result here is that the fully protonated form of this molecule has 1, 2 positive formal charges, so the net overall charge on this species is +2. These are the fully protonated forms, so they're the place you start with an acid-base reaction for either of these species. The side chain for aspartic acid has a pKa of 3.86, rather similar, just a little bit higher than the backbone carboxylic acid pKa's. And the side chain pKa for the histidine, that this is the ammonium or iminium form, and it has a pKa of 6.804 for turning into the imine form. Test yourself, please. Start with the fully protonated form here for aspartic acid, and go ahead and write the full and schematic forms of the reactions that this will undergo with water. Then label the isoelectric species. And I'd like you to think, what order do you need to lose these acidic groups? Remember the rule of thumb. You start with the most acidic group first and move up to the next acidic group and the least acidic group with the highest pKa. So pause the video and write out all those reactions. This is probably getting more challenging now. Here's the set of reactions I would expect you to draw. First, the molecule itself in the fully protonated form with its positive charge because there's an ammonium ion and two neutral carboxylic acids, all the acid forms of all the groups in the molecule. That will lose one H+, and the one it will lose is governed by the one with the lowest pKa, which happens to be the backbone carboxylic acid. So that turns into a carboxylate ion, and the other two come along exactly as they were. This species then can lose another H+, and the one that leaves will be governed by the next lowest pKa, and the next lowest pKa is for the side chain carboxylic acid. So that will lose that H+ to form the carboxylate ion. Of course, the carboxylate ion that we already had gets brought along, as does the ammonium ion that we still had from the previous step. So the only thing that happened between form B here and form C is that we lost the side chains H+. And in the last step, with the highest pKa, we'll lose the ammonium ion H+ to form the amine. The charges went from a plus charge for the fully protonated form to a neutral charge here in the betaine form, to a -1 charge because we have two minuses and one plus adding up to the overall minus, to a 2- charge with the two negative charges and a neutral now for the conjugate base form of the ammonium ion. This is the amine instead. A, B, C, D, and you should be able to identify that the neutral, even though there's a plus and a minus, the overall charge here is neutral, identifies B as the isoelectric species for aspartic acid. Go ahead and test yourself again with histidine. Label the isoelectric species, write those full and schematic forms of the reactions. Just a note that on the previous slide, I didn't write out the full forms of the reactions, but I do expect you to do that for practice with several of these, where you react them with water and make H3O+. But you can write the schematic forms on the exams in our class. For histidine, you can start with the information that the pKa for the ammonium hydrogen is 9.17. For the alpha carboxylic acid in the backbone, it's 1.82. And for the side chain, iminium ion, it's 6.04. I'm not going to show you the answer to this, but please stop the video and write out your work and bring that to class. Here's another activity I'd like you to do. I'd like you to go through each of these amino acids, this isn't all of them, but it's almost all of them, and identify the side chain, predict whether the side chain will be a weak acid, a weak base, or neither. In other words, a molecule, like it was for alanine. So pause the video and do your best looking at the structures of these to decide what each of them will be. Perhaps you saw some patterns here. Alanine, with its simple methyl group, would just be a molecule. It has no acid or base heteroatoms in it. Aspartic acid, as we just saw, is a weak acid. It has a carboxylic acid group. Leucine has no functional groups. It's just a carbon framework, so it's a molecule. Glutamic acid, like aspartic acid, will be a weak acid. Glutamine has an amide group, and you may not know what amides do as acid or bases. They don't do anything. They aren't weak acids and they aren't weak bases in aqueous solutions. So that amide on the side chain of glutamine will just act as a molecule. Histidine we saw acts as a weak base. Valine with no functional groups will be a molecule. Cysteine, I'm curious to know what you predicted for cysteine. It doesn't have a carboxylic acid, it has a thiol group, and you have no reason to know whether that's a weak acid, a weak base, or a molecule. You perhaps predicted it as a molecule, but it turns out to be a weak acid. So this is one that would have been hard to predict, but I wanted to call your attention to it because I think by looking at the structures, you can get most of them right, but there's a couple that are going to be puzzles, and I want you to know what those are. By comparison with glutamine, I think you could probably predict that asparagine will be a molecule. That amide doesn't have any acid or base properties. Serine is an alcohol, and I told you in class that the alcohols are molecules, that they don't have acid or base properties. Threonine, likewise, would be a molecule. It's an alcohol functional group. But tyrosine, another alcohol on a phenyl ring, turns out to be a weak acid. I think that would be hard to predict. So again, I'm calling your attention to the ones that don't follow the simple structural rules. Lysine has an amine group, and that's a weak base. Arginine has a complex guanine group, and it also turns out to be a weak base. Phenylalanine has no functional groups other than the benzene ring, is a molecule. Proline, that just is a simple carbon side chain, is a molecule. Methionine, now that you found out cysteine is a weak acid, you may be wondering about methionine, but methionine doesn't have an H on the sulfur, it has a CH3, and so that turns out to be a molecule as well. There's no H plus that can do anything in that molecule. And tryptophan, also potentially confusing, has an amine in it, but does not turn out to be a weak base. It turns out to be a molecule. So I think that cysteine, tryptophan, and tyrosine are the most puzzling of our amino acids. You may be feeling a certain amount of despair right now, like, can you ever predict anything? But there are some big patterns. The amino acids that you learned as the nonpolar amino acids are all side chains that are molecules. They don't have acidic or basic properties. So that's an easy way to remember that tryptophan doesn't have any acid or base properties. The amino acids that are charged all have side chains that are either weak acids or weak bases. Aspartic acid and glutamic acid are obviously acids, and histidine, lysine, and arginine are weak bases. So they exist with in their conjugate acid forms in the fully protonated state. Here are pictures of asp and glue, fully protonated. The ammonium, the carboxylic acid, and the other carboxylic acid in the side chain, all in their conjugate acid forms. Here are histidine and lysine in their fully protonated forms, carboxylic acid, ammonium, and the iminium here, the extra hydrogen on the histidine ring, and the ammonium here, the amine that's at the end of lysine in its protonated form is an ammonium group. So we have an ammonium, another ammonium, and a carboxylic acid. The fully protonated form here is +2, as it is with histidine. And arginine's fully protonated form looks similar. The carboxylic acid, the ammonium, and down here, the guanidinium group, you have 4 bonds to this nitrogen and a positive formal charge. So the overall charge is +2. The only ones that are difficult are the polar amino acids, because some of those behave as you'd expect them to. Serine, threonine, asparagine, and glutamine don't have any acid or base properties, but cysteine and tyrosine both have acidic properties. Cysteine, remember, looks like this. Here's its fully protonated carboxylic acid and ammonium ion in the backbone, and when it's fully protonated, it looks just the way you memorize the side chain, CH2SH. So it's this H that will end up leaving when it undergoes an acid-base reaction. And for tyrosine, again, carboxylic acid, ammonium group, and the side chain is the neutral one that you memorized. This H ends up leaving as an H plus and leaves this whole ring with a minus formal charge. Very, very difficult to predict with what you guys know right now about functional groups and the way compounds behave. But you need to memorize it because it's important for their reactivity. Luckily, we give you a handout that helps with all this. Here is a handout that you'll have on the exams that with the amino acid pKa values and side chain identities. So for alanine, we follow across. Here you see the pKa value for its backbone carboxylic acid group, its backbone ammonium group, and its side chain is a simple CH3, so it doesn't have any pKa for that side chain. That hydrogen is not acidic at all, doesn't leave. This is not a minus charge. This is where that side chain attaches to the main backbone of the amino acid. Arginine has its backbone carboxylic acid at 2.17 for a pKa, its ammonium group at 9.04, and its side chain pKa at 12.48. So that means here's where it attaches to the backbone. So this is CH2, 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 NH, C double bond NH2. This is the extra proton in the fully protonated form of the side chain. In all cases, this column shows you the fully protonated form of the side chain. So this is where you'd start if you're going to draw reactions that involve this molecule. You'd start with a fully protonated form, and that H wouldn't leave until a pKa of 12.48. Aspartic acid, backbone carboxylic acid 2.09, backbone ammonium at 9.82, and 3.86 for its side chain. This is the acidic proton in the side chain that will leave. Cysteine, 1.71, 10.78, and its side chain has a pKa of 8.33. Again, it's this H plus that will leave, and what will be left is this attached to the backbone, CH2S with a minus formal charge when that H plus leaves. Here's the rest of your table, and I'm going to leave you with another test yourself to bring to class. Write the full and schematic forms of the reactions for both cysteine and tyrosine. You'll need to use that handout that we just went through. Be sure to label the isoelectric species and bring your written out work to class. Here's what the sample exam question looks like for this outcome. We'll ask you what the predominant form of some amino acid is at some specified pH. And after this video, you should be able to do this first bullet. Draw the structures of all species present in aqueous solution, show which ones are in equilibrium with each other, and label the equilibrium arrows with the appropriate pKa's. You should also be able to pick out the isoelectric species. In class, we'll go through how to draw the abundance diagram, calculate the pH at each maximum, and label the isoelectric point, not just the isoelectric species. And then, much as we did with outcome 12, you'll be able to determine the most abundant form just by reading the graph at the given pH.